Hi, everyone. Well, we wanted to put together a very short slide deck to go over the technique of photoconversion since it's used in the Kelly et al. Paper that we'll be discussing in class 5 as well as class 6. So what is photoconversion and what can it be used for and how did they use it in the paper? So first, just the general outline on the different kinds of fluorescent proteins and photoconversion techniques that can be used. So these are fluorescent proteins that have modifications to them that allow for certain manipulations. One is photoactivation. So in this case, it actually doesn't have a fluorescence, it's in its off state. And upon illuminating it with a certain wavelength of light, it then begins to fluoresce in GFP. Another example is photo switching, which goes from an off state, so no fluorescence, to an on state with fluorescence, and back and forth with two different wavelengths of light. So for example, you might irradiate it with 488 nanometers of light to create a GFP molecule that fluoresces. And then when you use 405 nanometers of light, it turns it back off into an off state with no fluorescence. And the example that I'll be talking about a little bit more today is photoconversion, which is a fluorescent molecule that is first generally GFP fluorescing, so fluoresces in the GFP channel, and then when you illuminate it with light, it switches into the red channel. So this is not generally reversible. It's a one-way photoconversion. And the ways we would want to use photoconversion is if you have sort of a uniform or a ubiquitous expression of fluorescence. So that is shown here where you have an example where there are a bunch of GFP structures that are positive for GFP. And when you look at the next time point, you see that many of them have moved. But you can't actually say which one moved to which location. So is this GFP positive molecule now? Before, was it this one, or was it this one that moved in, or is it this one that moved over? Because all of these different GFP molecules are uniformly distributed in the cell and all sort of moving at different rates at different locations. However, if this molecule was tagged with dendra-2, which is the photoconvertible protein that is used in the paper, I could then mark a subset of those molecules with red, right? So convert them from green to red. And now when I look at the next time frame, I'm only marking a subset to then follow over time. So the most likely scenario then is that this molecule was this in the previous time frame, and this one was this one in the previous time frame. So photoconversion is a really powerful technique to mark a region of a uniformly GFP labeled tissue to be able to ask how does that specific region move over time. So in the paper, they use it to mark extracellular matrix to ask how the extracellular matrix is moved or degraded. So I hope this gives you enough information to be able to understand the paper or this technique in the paper.